So that covers the colloid osmotic pressure. Moving to the effective oncotic pressure, it is the difference between the higher oncotic pressure of plasma and the lower oncotic pressure of the interstitial fluid, and is the force that tends to draw the fluid into the vessels. So when there is a higher and a lower concentration or the lower pressures that has taken place, usually from high pressure to low pressure transition takes place. So this pressure is called as effective oncotic pressure. So difference in the pressures will lead to the draw of fluid into the vessels. Going to the next one, which is hydrostatic pressure. It is a capillary blood pressure that is present. So the pressure gradient depends upon the two ends of the capillary loop. One is higher at the arteriolar end, and the other is lower at the venular end. So near the arteriolar end, there is average of 32 mmHg of pressure, and near the venular end, 12 mmHg of pressure is observed. So the tissue tension is the hydrostatic pressure of the interstitial fluid and is lower than the hydrostatic pressure in the capillary at the other end. So the effective hydrostatic pressure is the difference between the higher and the lower higher hydrostatic pressure in the capillary and the lower tissue tension. It is a force that drives the fluid from capillary wall to the interstitial space. So you can, as the points say, that there is a higher pressure in the in the capillary wall. So when there is a pressure inside the capillary wall, there is a gradual tendency of the pressure that moves from inner capillary wall into the interstitial place, where there is lesser pressure. So that is called as hydrostatic pressure. Going to the normal fluid exchanges that are taking place inside the body compartments are at the arterial end of the capillary. So there is a balance between hydrostatic and the plasma oncotic pressures. So when there is this balance, the hydrostatic pressure of 7 mmHg, which is outward driving force, so that the small quantity of fluid and solute leaving the vessel to enter the interstitial space. So this minimum of 7 mmHg hydrostatic pressure drives the force of moving the smallest quantity of fluid into the interstitial space from the vessel. At the venular end, the balance between the hydrostatic and the plasma oncotic pressure is inward driving force. So from the interstitium, the fluid enters into the venular end of the capillary. So there is opposite kind of fluid exchange that is taking place near the venular end of the capillary, and the fluid that is left after exchanging across capillary wall escapes into the lymphatic. So whatever the fluid that is being left out after the exchange is taking place is usually sent to the lymphatic, which is finally drained into the venous system itself, venous circulation itself. Going to the tissue factors, which are normally small and insignificant force opposing the plasma hydrostatic pressure and capillary hydrostatic pressures respectively. So there are various tissue factors that might affect the hydrostatic pressure as well as the plasma hydrostatic capillary as well as plasma hydrostatic pressures. So certain tissue factors which are determining these two forces or the pressures are oncotic pressure of interstitial fluid and the tissue tensions. So that covers the normal fluid exchange. So this is how homeostasis is maintained inside a human body. So to wrap up the session of this video, I'd like to tell that the introductory part, which involved the Claude Bernard, who first coined the term internal environment. Moving to the next part, which is the mechanism of homeostasis, which is by maintaining the internal environment in a balanced state. uh, Usually homeostasis is maintained by exchange of fluids, electrolytes, nutrient metabolites, etc. And there are two normal composition components that are present inside human body, which is water and electrolyte. Water is a principal part of human body, and it comprises about 60% on the human body weight. um, And the total body water can be divided into intracellular compartment and extracellular compartment. So intracellular is inside the cell and extracellular is outside the fluid, outside the cell. And the extracellular fluid compartment can be further divided into four subdivisions, which are interstitial fluid, intravascular fluid, as well as mesenchymal tissue fluid and transcellular fluid component. So that covers the water components. Now moving to the electrolytes, the intracellular fluid contains major cations as potassium and magnesium, and major anions as phosphate and protein. In extracellular fluid, the predominant cation is sodium, and principal anion is the chloride. Moving to the major function of electrolyte, it helps in maintenance of acid-base balance. It helps in maintenance of proper osmolality and volume of body fluids, and it helps in the concentration of certain electrolytes, which is important in the specific physiologic function. And we also studied about the Gibbs-Donan equilibrium, uh, how the water is being eliminated from the body, and we study about acid base balance, the buffer system, pulmonary mechanism, and renal mechanism. The pressure gradients and fluid exchange was also one of the topic. So this covers the homeostasis part of the physiology. Hope you like the video. If you do like the content that is being put in our channel, just like, share and subscribe to the channel to have more updates on it. Thank you.